तो गाइस वेलकम बैक टू पेज रिप्रेप चैनल। सो बेसिकली जो हम DHR ICMR BRET JRF के एग्जाम्स बहुत नियर है, लाइक ऑलमोस्ट वी हैव ओनली 1 week to go. So before that we have to rapidly revise everything, and specifically we have been focusing on part B for rapid revision because there are some units which are very crucial and which are not actually available that much online. So basically one of that is biotechnology process and applications, which we have told that we will bring you this completely. So the main focus of this unit will be in vitro techniques, DNA technology, computational biology, spectroscopy, microscopy. These topics are very, very important concerning biotechnology process and applications. So if you see into a deep dive into the details of this, the contents will be um, majorly like DNA, RNA isolation, southern blotting, Western blotting, ELISA, radioimmunoassay, immunohistochemistry, or for sequencing methods, whole genome sequencing, chip sequencing, PCR, types of PCR, बायोइन्फॉर्मेटिक्स, हाइब्रिडोमा टेक्नोलॉजी, क्रिस्पर कैस्पर, बहुत सारे ऐसे टॉपिक्स है जो मैंने इस पूरे इसमें बहुत शॉर्ट एंड क्रिस्प वे में एक्सप्लेन कर लिया है, तो डोंट मिस इट एनीथिंग. एंड अमंग दिस द वी हैव आल्सो आइडेंटिफाइड द मोस्ट इंपॉर्टेंट टॉपिक्स लाइक इम्यूनोहिस्टोकेमिस्ट्री हो गया, जीनोमिक cDNA लाइब्रेरीज हो गया, सीक्वेंसिंग हो गया, PCR बेस्ड मार्कर्स हो गया, प्रिंसिपल्स ऑफ माइक्रोस्कोपी एंड स्पेक्ट्रोस्कोपी हो गया, क्रिस्पर कैस्पर हो गया. These are very, very, very important topics that you all have to focus. So coming to let's start the lecture. पहला जो कि हम देखने वाले हैं DNA वर्सेस RNA आइसोलेशन, विच इज़ वेरी 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 बेसिक थिंग इन बायोटेक्नोलॉजी प्रोसेस एंड एप्लीकेशन, और टेक्निक्स का यूनिट भी आप अगर लेंगे ओवरऑल, तो बेसिकली द DNA वर्सेस RNA आइसोलेशन में पहला DNA आइसोलेशन हम देखेंगे. तो अगर वी डू द प्रोसेस ऑफ DNA आइसोलेशन इन pH अल्कलाइन pH, दैट इज pH 8.0, और जो रिएजेंट हम यूज़ करते हैं टू लाइसिस द सेल्स, दैट इज CTAB और HTF फॉर लाइसिस, एंड प्रेसिपिटेशन के लिए जो हम यूज़ करेंगे दैट इज आइसोप्रोपेनॉल और एथेनॉल, और जो स्टेबिलिटी ऑफ DNA है वो स्टेबल होता है. एंड कमिंग टू RNA आइसोलेशन का हम करते हैं एसिडिक pH में, दैट इज 4.0 to 5.0, एंड द रिएजेंट दैट वी यूज फॉर RNA आइसोलेशन इज ट्राइजोल और गुआनिडियम आइसोथायोसाइनेट. तो बेसिकली और यहाँ पे प्रेसिपिटेशन के लिए हम लिथियम क्लोराइड यूज करते हैं. सो लिथियम क्लोराइड इज मोर स्पेसिफिकली फॉर सिंगल स्ट्रैंड RNA, सो यू शुड नोट इट इन योर नोट्स. और जो RNA का स्टेबिलिटी होता है दैट इज इट इज मोर अनस्टेबल. So coming to the ratio of the DNA or RNA का, so the ratio that we use to get pure DNA is done by a ratio of A260 by A280. तो इसका रेशियो हम जब निकालते हैं वैल्यूज़ का, तो इट शुड बी पर प्योर DNA इट शुड बी अराउंड 1.8, तब वो प्योर DNA होता है, और पर प्योर RNA इट शुड बी अराउंड 2.0, और अगर इट इज़ अराउंड 1.6 इट इज़ प्रोटीन कंटामिनेशन. So let's come to next topic, that is isolation and detection of DNA, RNA and protein by southern western blotting. So आप सबको बताया like before PCR और whatever technique southern blotting था, और फिर RNA के लिए northern blotting था, और western blotting के लिए तो western blotting is the gold standard of protein identification. So when coming to southern blotting, the target molecule is DNA. इसमें membranes हम nitrocellulose और nylon membrane यूज करते हैं, और separation technique इसका है कि agarose or polyacromate process. The probes that we use in this is nucleic acid probes having a homologous sequence with the target molecules, probably radiolabeled enzymes that we use. This is the main reason that we have left this apart, because we use radiolabeled enzymes and it is cytotoxic to use. So we should very carefully we should use. The detection methods are chemiluminescence, colorimetry, and X-ray film. Blotting techniques are capillary transfer we do. Applications are used in forensic and study of DNA mutation. Western blotting is most famous technique, and the target molecule of this is the protein. So membranes used are nitrocellulose PVDF membrane. PVDF membrane is most heavily used like for most molecules which have protein molecules which have larger kilodaltons of weight, more molecular weight. Those for that PVDF membrane is used, and for nitrocellulose, like we have more like below 10 kD protein molecule, then you can use nitrocellulose. So suppression technique is Phage. The probe used a primary antibody and then continued with secondary antibody. The probe labeled used is enzyme. For detection methods are infrared imaging system, OLED CCD or LED imaging system. Blotting techniques, electroblotting we do. Applications is used in identifying specific antigens of proteins from different biological samples. Coming to northern blot, that is RNA, that is the target molecule. So we use here diazobenzomethyl or nylon membrane. The separation technique is agarose gel electrophoresis. Probes used are DNA, RNA and oligonucleotides. Probe labeled used at radiolabeled enzyme. So same here, like southern blotting, chemiluminescence, calorimetry and expressions can be detection methods. Blotting technique is capillary. And these applications are used in gene expression studies and helps in disease diagnosis. Coming to next step now, the most important technique among all, that is ELISA, that is enzyme linked immunosorbent assay. Like most of you have been familiar with it. So there are four types of ELISA, that is direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Direct ELISA involves only single antibody that is directly probed, which has radio-labeled enzyme, like radio enzyme that can give reaction and substrate is reacted. And indirect ELISA, it is primary antibody and secondary antibody both are involved. Sandwich ELISA is like it is the antigen is sandwiched between the antibodies. In competitive ELISA, there are two antibodies that are competitive for the antigen. So, in direct ELISA, antigen is detected usually when the specific immune response need to be analyzed quickly. Example, epitope mapping, we use direct ELISA. And indirect ELISA, antibody detection is done here, determining antibody concentration in serum. It is used for HIV screening. And sandwich ELISA, here antigen quantitation is done, which is highly specific, best for complex samples, that is blood, serum, and because the purification of antigen isn't needed. And sandwich ELISA example is pregnancy test. And now competitive ELISA, so small molecule detection is done in competitive ELISA. So usually when an antigen has only one epitope or it is too small for sandwich ELISA. So the most example, if you take, hormones have been detected by using this. Hormone concentrations have been detected. So you should note the notes also, or take notes also. If you want to take screenshots, you can also take screenshots. So among all these different types of ELISA, direct ELISA is most rarely used because it is way too far costly than any of these ELISA. So you should also make note of that. So coming to the next part, that is radioimmunoassay. So radioimmunoassay is one of the immunoassay techniques. So radioimmunoassay is an in vitro assay. It calculates the presence of an antigen with very high sensitivity. So mainly any biological product or substance for which a specific antibody exists can be measured even in minute concentrations. Radioimmunoassay was the first immunoassay procedure developed to analyze picomolar and nanomolar concentration of hormones in biology. See, in very simple language or layman language, when you have a very low amount of concentrations of antigens per detection, then this is the best technique to use. Like if someone has been detected with some disease, like if someone has typhoid and salmonella typhi is the like vector, so you should have the specific antigens generated in your body, so that is taken out, and you will also have radio labeled antigens, so you both are being mixed and even for the detection. So for example, if you have more number of radio labeled antigens being detected, then you have no disease. Okay, so same vice versa, if you have more number of very less number of radio labeled antigens detected, then you have the disease of that specific thing. So coming to the next topic, another immunoassay technique, that is immunohistochemistry. So immunohistochemistry is basically a combination of three things, that is immune, histo, and chemistry. Immune means you have the antibody involvement here, and histo means you have the tissues to which the antibodies will be binding, and chemistry means antibody antigen reaction will take place here. So this is the main concept of this thing. So primarily tissue preparation is done, like this is a tissue. In tissue only we will see the immunohistochemistry, and in other things also we can see, that is the cells also been sliced and seen like cell morphology, and also we can see in this. So primarily tissue preparation is done, and then the paraffinization is done. Like we will have the paraffin wax soluble, and then it is cut in the microtome machine that you can see in the machine, that it will slice into very thin, like very into 1 nanometer of scale it will cut. So after inactivation of the tissue is done, antigen retrieval is done, blocking is done, primary antibody incubation will be given, secondary antibody incubation will be done, and then staining will be done. So at the final you will have an image. You have the A1, A2, A3, B1, B2, B3, you can see now. So this is the image that you will get finally. So basically you will give antibody, and it will go and bind to the antigen. So the amount of expression of some specific protein that you can see by this technique. So I want to remind you all that again we have initiated this concept of rapid revision for to revise everything very quickly. So we have already published various other videos on biosafety, basic physiology, genetics and genomics, evolution in original life. Everything has been out. These are also very, very important units. You can also see these are also like under 50 minutes. These videos are, you can have a very quick revision. Very short points or main points have been given. Every link of these videos, if you have missed out, we have provided in the description. You can go and check it out. So coming to the next topic, we are moving. Now we have come to the next topic. That is this. Now we will begin DNA technology, recombinant DNA technology topics. So basically, the first topic will be restriction enzymes. So restriction enzymes are like these are three types in nature. So the classified into type 1, type 2, type 3 restriction enzymes. So let us see a talk about how much this restriction enzyme that we have like made for short and crisp. So these are the very important points. So particularly in type 1, the activity is bifunctional enzyme with endonuclease and methylase activity. The subunits are of three subunits in type 1. The cofactors that are used for the type 1 are ATP, magnesium, and as adenosine methionine. The cleavage site of type 1 restriction enzymes are random up to 1000 base pairs away from the restriction site. In type 2, a unifunctional enzyme either with endonuclease or methylase activity is there. There are two identical subunits in type 2. Cofactor used here is only Mg plus 2, and the cleavage site is at or near recognition sequences of in a DNA. Okay. So coming to type 3, it is a bifunctional enzyme with endonuclease and methylase activity. There are two subunits in this restriction enzyme also. And next, the cofactor is ATP and Mg plus 2. Here also the cofactors are ATP and Mg plus 2. And the cleavage site is 24 to 26 base pair downstream from recognition sequence. So this is all about the types and restriction enzymes, and examples of restriction enzymes are listed in the tabular form. You can see that. You can like take a screenshot of it. So basically, overall, restriction enzyme processes a biochemical process where an enzyme speeds up a specific chemical reaction conversion of a substrate into product. So these are uh, molecular scissors that are found in bacteria, cutting DNA at specific recognition sites, crucial for defense against viruses and essential tools in genetic engineering and manipulating DNA, creating recombinant DNA, and cloning by producing sticky and blunt ends. So the ends that are generated by these are sticky ends and blunt ends. Blunt ends is that cutting exactly at the center and differentiating both the DNA strand, and blunt ends is that you will have two cuts and you will have so a specific part of the one segment of the DNA is lifted from one segment like that. So coming to next topic, cloning vectors. So cloning vectors. A cloning vector is a small self-replicating DNA molecule like plasmid or virus DNA used in genetic engineering to carry and multiply a foreign DNA fragment. So inside a host cell, creating many identical copies. The key features that include in a uh, cloning vector is that origin of replication for self-propagation, a multiple cloning site with restriction in sites for inserting DNA, and a selectable marker like an antibiotic resistance gene. So successfully transforming cells. So there are various types of cloning vectors. Cloning vectors. So the specific type of vectors capacity have been listed here, which are most important for the exam point of view. So plasmid is the insertion size is less than 10 kilobase pairs. Host is bacteria. Phase lambda insert size is 10 to 25 kilobase pairs. Plasmid is 30 to 45 kilobase pairs. Bacterial artificial chromosome is 100 to 300 kb, and yeast artificial chromosome is 200 to 2000 kb. The yeast artificial chromosome host is yeast. And coming to genomic versus cDNA library. So both these libraries are nothing but having copies of genetic information. So specifically, genomic library contains introns plus exons plus regulatory sequences, same for all tissues of an organism. Whereas in cDNA library, that is made from mRNA. So cDNA libraries are made from mRNA, that is after expression. So these mRNAs are taken, and these only have exons, and these have only tissue specific expressions. Also, these are used for seeing the tissue specific expressions, not overall whole genome. Okay, so coming to next topic, here I have summarized the three main topics in DNA technology, that is site-directed mutagenesis, yeast to hybrid system, and phage display. So these three topics are also very important. So I have very much crisply and shortly cut it down into a single table. So coming to site-directed mutagenesis, the primary goal of the site-directed mutagenesis is modify a protein's DNA sequence at a specific time. And the basic concept is surgical editing of DNA. The host system that we use is in vitro. You can use in PCR, and also we can do this experiment in E. coli. Input we have to give is a non-genome plasmid, and you will get a DNA sequence change. Advantage of this is high precision for functional studies, and limitations are low throughput, needs prior knowledge. And in each two hybrid system, the primary goal is identify protein-protein interactions. Basic concept is matchmaking inside a yeast cell. The host system you can use in here is Saccharomyces cerevisiae. Input is you have to give two proteins, and readout you will get activation of a reporter gene. Key advantage is detects interactions in a living eukaryotic cell. Key limitation is high false positive rate is there. In Phage display, the primary goal is select high affinity parameters from a library. The basic concept is fishing using viral successes. Host system is bacteriophage, usually M13 and E. coli. Input is a library of gene variants you put it, and readout you will get bio panning, binding or selection. And key advantage is extremely high throughput and limitations limited to proteins that fold in bacteria. Inside the bacteria, which proteins fold, those are only used here. So next topic is sequencing. So sequencing, when coming to we have DNA sequencing. So in most early days we have Sanger method that uses deoxynucleotides as chain terminators which lack 3 OH group. And coming to next generation sequencing, we have pyrosequencing, Illumina and ion torrent. In pyrosequencing, that detects pyrophosphate release via luciferase light, and in Illumina bridge amplification is done and reversible terminators are used. In ion torrent, it detects the H plus ion release, the pH change due to pH change. So coming to the sequencing, there are first generation, second generation, and third generation sequencing types. First generation is Sanger, second generation is Illumina, and third generation nanopore, that is most advanced generation sequencing. So you can see this and take a screenshot of this also, and further you can read about this in online and YouTube. And coming to next topic is whole genome sequencing. So whole genome sequencing is sequencing of the complete genome of a person. Like if a newborn human being is there, like baby is there, you can do the whole genome sequencing, and priorly you can map his genome to a reference genome and see whether he is prevalent to any kind of genetic disorders like cancer, diabetes. These are also associated with genetics, na? So those kind of things you can do with whole genome sequencing. So here the reference genome is taken, and also individual genome is taken, and they are broken into many small fragments and cloned, and then the broken individual clones into small fragments are then mapped according to a reference genome. So the reference genome is primarily mapped, and then taking the reference genome into this context of the individual gene, they both are aligned properly, and then you can see any SNPs like VNPs or whatever variants you found, then you can categorize them. Then you can categorize them into disease or non-disease, or what kind of prevalences that he has regarding something. So coming to next one, that is chip sequencing, that is chromatin immunoprecipitation sequencing. So this is a high throughput technology that is used in the lab. So basically, chip sequencing combines chromatin immunoprecipitation with massively parallel DNA sequencing to identify the binding sites of DNA associated proteins. So the major goal of this method is to find which proteins are binded to DNA, and is there any association between them or not, to check this sequencing is related. So the primary step is you will give formaldehyde to the cells in the cell culture, and the proteins which are associated with the DNA bind at that step. In a particular phase you will give that, and then DNA protein cross-linking happens, and then cell lysis you will do, and sonication to share DNA antibodies in molecules to beads bind to DNA binding protein, and you can wash to remove non-target proteins in DNA, and then a reverse cross-linking of digestion proteins, purify and amplify DNA, and then you can take the downstream applications. So you can do qPCR and see the readings. Okay, now coming to next topic, that is polymerase chain reaction. So PCR, it is a fundamental lab technique that rapidly makes millions to billions copies of a specific DNA segment, allowing scientists to study it in detail, diagnose disease, or analyze genetic material from tiny samples using cycles of heating and cooling with primers and a DNA polymerase enzyme. So it is developed by Carey Mullins in 1983. It's essential for DNA amplification in research, forensics, and diagnostics like detecting viruses. So how it works? So the primary step of this process is denaturation. Heat separates the double-stranded DNA into single strands. And the second step is annealing. The mixture cools, allowing primers that are short DNA pieces to bind to the target DNA sequence. And then extension. A heat-stable DNA polymerase like tag polymerase adds nucleotides to build new complementary DNA strands starting from the primers. And then last is cycling. These steps are repeated exponentially, increasing the number of targeted DNA copies. So this is the basic principle of the whole polymerase chain reaction. And then next move to the next different types of PCR. So there are various types of PCR that have been developed during the course of the time according to the experiment requirement. So the first one is standard endpoint PCR, that is basic three-step cycle: denature, anneal, extend, and best used for is routine cloning, sequencing, and simple SNO detection. Next one is RT-PCR, that is reverse transcription enzyme is used in this. It uses reverse transcriptase to turn RNA into cDNA first. So it is best used for detection of RNA viruses like COVID-19 or gene expression. This is the mostly used method during the COVID to detect whether a person has COVID or not. And then qPCR, that is quantitative or real-time PCR. It uses fluorescent dyes to monitor amplification as it happens. It is like you have the real-time visual of a gene expression. For that, this thing is used, and to it is best used for the measuring the exact amount of DNA or RNA in a sample. So next is multiplex PCR. It uses multiple set points of primers in one single tube, testing for several different pathogens at the same time. So if you have a mixture of bacteria inside what we say sample, you want to have a different kind of primers, and you want to do all the experiments at the same in a same experiment setup, then you can use multiplex PCR. And then nested PCR, it uses two rounds of PCR with two sets of primer, inner and outer. Best used for is increasing sensitivity when DNA is very scarce or contaminated. And then digital PCR, that is digital PCR. So key mechanism of this is partitions the sample into thousands of tiny droplets, and it is best used for absolute quantification of rare mutations. And then the final one is touchdown PCR, gradually lowers the annealing temperature in each cycle, and it is best used for reducing non-specific binding, getting cleaner result. So these are also very very important points you should take care of. And coming to next segment of the PCR is PCR based markers. So there are different types of markers that are PCR based. So like RAPD, SSR, SNPs, ISSR, SCARs, like this. So RAPD is a random amplified polymorphic DNA. These are single short and base pair random primers. So inheritance type is dominant, and DNA sequence knowledge is not yet known. The main use is diversity studies in species with no known sequences. And the second type of marker is SSRs, that is simple sequence repeats. These are microsatellites, and these are specific primers flanking a repeat. In inheritance type is codominant, can tell heterozygotes. DNA sequence knowledge is less known, and main use is paternity testing, mapping, and population genetics. It is mostly used. And the SNP, that is single nucleotide polymorphism. These are sequence specific, single base changing, and inheritance type is codominant. DNA sequence knowledge is, yes, you know that. And this main use is high throughput genome wide association studies, that is GWAS studies, that it is used for. And then next one is ISSRs, that is inter simple sequence repeats. These primer types are one primer based on an SSR motif, and it has a dominant inheritance, and the sequence knowledge is not known. And the main use is genetic fingerprinting and phylogenetic studies. And the final one is CARs, that is sequence characterized amplified regions. These are longer specific primers derived from RAPD, that is random amplified polymorphic DNA. And usually these are codominant inheritance showing, and the sequence knowledge is known. And these are main use is reliable markers assisted selection in breeding. Coming to next segment, it is DNA fingerprinting. So DNA fingerprinting is a technique based on DNA polymorphism, specifically utilizing variable number tandem repeats, which fall under the category of the mini satellite DNA. So the standard procedure of this begins with the isolation of genomic DNA from nucleated cells, not RBCs, followed by digestion with restriction endonucleases to create many kind of fragments. These fragments are then separated by size using agarose gel electrophoresis, and then transferred to a nitrocellulose or nylon membrane via southern blotting. The membrane is hybridized with a radioactive single-stranded DNA probe complementary to VNTR sequences, and the final unique pattern of bands is visualized to autoradiography, serving as a distinct identifier for every individual except monozygotic twins. So basically, this has a broad, what we say, broad application in forensics. Like if there is a crime happened and you have two suspects, and you have the main suspect's fingerprints on an object, and you want to see who is the culprit out of the two suspects that you have identified. So you can take the you can do DNA fingerprinting of the samples, and then you can see who is the main culprit. So this is the by far the main concept of the DNA fingerprinting. And the next topic now is in situ hybridization or fluorescence in situ hybridization. In situ hybridization is a powerful lab technique that uses labeled single-stranded DNA or RNA probes to find and visualize specific complementary DNA or RNA sequences directly within fixed cells or tissue sections. So, revealing where genes are active or located at a cellular level, essential for studying development, disease, and gene expression patterns using methods like FISH or SHISH, that is fluorescence in situ hybridization, or chromogenic in situ hybridization. So how it works? Generally, it has three steps, that is targeting, hybridization, and detection. So targeting means a synthetic nucleic acid probe that is short labeled DNA or RNA strand with a sequence complementary to the target gene sequence is created, and then the hybridization steps comes. The probe is applied to a thin slice of tissue or cells where it binds to its matching target DNA or RNA sequence, and then the final step is detection. A reporter molecule like a fluorescent dye or enzyme attached to the probe allows researcher to see where the binding occurred using microscopy. That is fluorescence or bright field you can use. So the main types of this fish is that these types of in situ hybridization are fish or fish that is chromogenic in situ hybridization. So fluorescently labeled probes visualized with a fluorescence microscope, and it is great for detecting chromosomal abnormalities, that is like example is Down syndrome or viral infections. Whereas in chromogenic in situ hybridization, to use probes with chromogenic like color producing molecules for detection under a standard bright field microscope, often combined with tissue morphology. So make this important point in your mind that fish is basically in fluorescence, and that chromogenic ones is color producing ones, and that is used in the bright field microscopes. So this can be a main question in ICMR also. So now let us start next topic, that is microscopy. So before starting microscopy, I would like to request you all to like and subscribe this channel, and please comment if you have any doubts regarding any topic. We will have a deep discussion or clarification in the comment section. And also please like and subscribe our channel. So and if you support us, we can bring more videos to this channel, and we can also grow as a community and help you succeed any kind of entrance exams in life sciences. So come on, let's get back to the topic, that is microscopy. So microscopy, if we see in a short and crispy way, I have combined every microscope and its principle in a short and crisp table. Like microscope type, if you see there is bright field microscope, the working principle is light passes directly through the sample. Contrast comes from light absorption by the stain, and in dark field, a central stop blocks direct light. Only scattered light from the specimen enters the lens. In phase contrast, converts phase shifts, invisible differences in refractive index into brightness differences. And coming to fluorescence, stroke shift takes place, that is absorbs light at one wavelength and emits at a longer wavelength. And coming to the next one, that is confocal microscopy, it uses a pinhole aperture to block out of focus light. And then electron microscopy, in this uses electron beams are used instead of light. So that is the principles and basics of the microscopy and types. So now the main thing, that is the differences, that is between light microscope and electron microscope. And if we take electron microscope, there are two types, the TEM and SEM, that is transmission electron microscope and scanning electron microscope. And then we have phase contrast and dark field microscope and their differences. So these are also very main important points. I suggest you to, I'm not going to explain this all again because it will become more lengthy. I request you all to take a screenshot of this and read for yourself and make notes of this. Okay, so next we will skip to the next topic, and the next topic is spectroscopy and its types. So in spectroscopy we have different types of spectroscopies. You have UV visible spectroscopy, and here the region used is UV region of 200 to 400 nanometers, and visible region is 400 to 700 nanometers. So the basic principle of UV visible spectroscopy is electronic transition, that is electrons jump to higher energy orbitals. And biological application of this UV visible spectroscopy is quantification of DNA and protein. So and the next one is fluorescence spectroscopy. So in fluorescence spectroscopy, you can have UV visible same thing, and the basic principle is emission of light at a longer wavelength after absorption. And the biological application of this is protein folding. You can see binding studies, GFP localization, you can all see this thing. And then the next one is circular dichromism. Here the far UV wavelength is used, that is 170 to 250 nanometers, and the basic principle is differential absorption of circularly polarized light. And the biological application of this secondary structure determination is done, that is alpha helix versus beta sheet. You can determine which kind of structure it has formed the protein in the protein. And the next one is infrared, that is IR or FTIR. Here the rays used is infrared rays, and the basic principle is vibrational transition, that is prone, stretch or bend. And the biological application is that identifying functional groups, that is monitoring ligand binding reactions. And the next one is NMR, most popular one, nuclear magnetic resonance, and here radio orbs are used. Basic principle is change in nuclear spin in a magnetic field. It is used to determine the 3D structure of proteins in solution, unlike X-ray, which needs crystals. And the next one is X-ray crystallography. X-rays are used. Diffraction of rays by electron clouds is basic principle, and the biological application is 3D structure at the atomic resolution. At the atomic level, you can have the resolution. And the final one is mass spectrometry. Here, the ions are bombarded together inside a like what we say vacuum, and there is one amount of pressure is used, and the separation is based on mass to charge ratio. And the biological application is molecular weight of a protein. Peptide sequencing is also can be done, and the proteomic studies it is used mostly. And now we have come to bioinformatics topic. So in bioinformatics, during the early days, there was no system and there was no amount of knowledge. Most of the work is done in wet lab. But now the increase in the amount of biological data has created a need for developing computational tools for managing and analyzing the data, which led to creation of a new field called bioinformatics. So bioinformatics is a rapidly growing field in biology that focuses on the development application of computational tools to analyze and interpret biological data. So bioinformatics software, tools, and databases are used to process, store, analyze, and interpret biological data. So in this lecture, we will have a very short snippet that is very much uh, um, uh, that is very much important in regarding of examination point of view. How much we need for the part B. Uh, if you see brief history, in 1965, Margaret Dieff created the Atlas of Protein Sequence Structure. In 1980, the EMBL Data Library created a catalog of biological data. In 1982, GenBank became a public database. In 2002, ARIA Express release. In 2005, the first high throughput sequencing system was used to sequence a full E. coli genome. And in present, many directions for multiomic data were created. So let's take to some databases. So coming to databases, biological databases, these are categorized into three types, that is primary, secondary, and derived databases. A primary database contains only sequence or structural information, whereas a secondary database is derived from the analysis or treatment of primary data, or second is called secondary database. It is very important for interfering protein function, like kind of data. And in derived, a derived database is a type of biological database that is created by analyzing and compiling information from other primary and secondary databases. A derived database contains processed, organized, and often annotated information. So if we see a brief what we say classification with examples, we have nucleotide, we have protein. If we see nucleotide, there are primary, secondary, tertiary databases. The primary are being EMBL, GenBank, DDBA, which have like raw data of the genetic material being deposited into that. And then the next one is specialized databases, that is Humin inherited disease database, gene expression OMBS, microarray database, array expression database. These are specialized database. When we come to protein, these are categorized into sequence, interaction, structure databases. For sequence databases, you have UniProt, protein information report, and SwissProt, which are all primary protein databases. And coming to the interaction databases, there are BioGrid and there are string databases where we can have data sets regarding protein-protein interaction. And coming to the structural wise, we have PDB, that is protein data bank, and then cath, and then scope. These are like cath is class, architecture, topology, homology, and the scope is structural classification of protein. So basically these are all main classifications of that. In nucleotide databases, the EMBL, GenBank, and DDBJ all are under the database of INSDS, that is International Nucleotide Sequence Database. And the whole we also have whole genome database, that is Ensemble, where you can get the whole genome sequences of various different kinds of organisms or whatever you want to do. If you study more about bioinformatics, you will get to know more application way of things regarding these things. So biological database. A database is an organized collection of related biological data that can be easily stored, accessed, and managed. If you say bibliographic database that contains article and research papers of different journals, if you can have example of PubMed, very popular and the main major source of publication database that we can get any article there. And then second one is sequence databases, contains protein and nucleotide sequence, GenBank, DDBJ, protein information report, all this. And the third one is structural database, contains 3D structure of proteins and nucleic acids, example is PDB. And then we also have metabolic database that contains data about various biological pathways, that is KGG, metapsych. These are the databases. You also have model organism database, contains in the biological data of studied model organism, that is examples are fibrase, RGD. We can find the Drosophila complete organism data here also. And then enzyme databases, example is Brenda. Then we have also disease database, you have chemical database that has data on several small organic molecules. And then microarray database, gene expression data from microarray experiments, GEO. And then also we have taxonomic database that provides information on species of animals, plants, and also catalog of life is an example of that database. Coming to next topic, we have BLAST, that is present in NCBI. BLAST is basic local alignment search tool. We have so it calculates similarity for biological sequences and produce and it produces local alignments. Only a portion of each sequence must be aligned. Uses statistical theory to determine if a match might have occurred by chance, like that. So basically in here, if you use a nucleotide BLAST, you will enter the nucleotide sequence and it will search for similar sequences which may have match or mismatch. So coming to the types of blast, we have blast P, blast N, blast X, T blast N, T blast X, blast 2CQ. So blast P is protein query against protein data bank, and blast T N is a DNA RNA query against DNA database. Blast X is 6' translation of DNA query against protein database. T blast N is protein query against 6' translation of DNA. This is very most important slide. Last year from this question has also came, so it is very much important. That's why I have included it. You can also take notes of this also and write it down, and you just have to read these types of bloods. These are the types that are present. Coming to the next important topic in bioinformatics is pairwise versus multiple sequence alignment. So pairwise alignment is done between two sequences, whereas multiple sequence alignment is done between three or more sequences. If you see further about the pairwise sequence alignment, the goal is to find regions of similarity between two sequences, and complexity is very low. The key output is percentage of identity and percentage of similarity to identify between the two sequences. And standard tools used to perform this pairwise sequence alignment is BLASTA, FASTA, EMBOSS, needle water, which algorithms are used in this. And then in multiple sequence element, the main goal is find evolutionary relationships or conserve domains across a family. The complexity here is very much high because in here there are three or more sequences are involved, and the key output is consensus sequence phylogenetic tree. Standard tools that we use here is cluster omega, muzzle, t coffee. These are the things that we use for this thing. And coming to next topic, we have hybridoma technology. So hybridoma technology is, if we say, basically like you have heard about monoclonal antibodies, that how monoclonal antibodies are like made. So in hybridoma technology, first if a mouse is there and you inject antigens into the mouse, antigen is uh, reciprocal to the kind of antibody that you are desired to get. So after antibodies are produced inside the mouse, we isolate the B cells, and from other side we get the myeloma cells, that are cancer cells which are HGPRT negative, and we begin to fuse these cells, and fusion of B lymphocytes and myeloma cells are done, and these are cultured in the hat media. Test each well for specific antibodies, screening of hybridoma cells, and then expansion of positive clones, and the clones which have the hat media ones, those like those proliferate, and then expansion of positive clones is done, and then in vitro propagation of positive clone is done, and then monoclonal antibodies are produced, and these monoclonal antibodies are further useful for clinical applications. So I want to tell a main point to you all, that is in hybridoma technology, the enzyme HGPRT, that is hypoxanthine guanine phosphoribosine transferase, is crucial for selecting desired hybrid cells. Myeloma cells used here lack functional HGPRT, making them die in hat medium, while normal B cells have it but die quickly, leaving only the hybrid cells, that is fused ones, that is B cells and myeloma cells, to survive and produce specific monoclonal antibodies. So that is the main concept of this thing. So coming to the next one, we have CRISPR-Cas9, which is very most important topic of all things. So the origin and function of it is the adaptive immune system of bacteria, specifically Streptococcus pyogenes, used to defend against attacking bacteriophages by mimicking the DNA. So the full form of CRISPR-Cas9 is clustered, regularly interspaced, short palindromic repeats. Coming to key components, Cas9, CRISPR-associated protein 9, the endonuclease enzyme scissor that cuts the DNA, which main role is cutting the DNA, and then guided RNA, a synthetic RNA that guides Cas9 to the specific target sequence. The sgRNA, if you see in an exam point of question in nature, bacteria use two RNAs, that is CRRNA contains the target code, and tracer RNA holds the structure. In the lab, these are fused into a single molecule called single guided RNA. So the tracer RNA is the typical sgRNA, single guided RNA, that is in the bacterial system, and the difference between that and the thing that we use in the lab is scientists use a specific one for the desired experiment that mimic the nature of the tracer RNA. And coming to next thing is PAM sequence, that is Cas9 cannot cut unless it finds a specific tag called PAM, protospacer addition motif, next to the target. For SP-Cas9, the PAM sequence is FIDAS and GG. It must be on that non-targeted strand immediately downstream of the target. So basically what happens is when a bacteriophage infects a bacteria, there is a what we say, a sequence called protospacer sequence. So that protospacer sequence is integrated into the bacterial genome between the repeats. So after that, the translation takes place after after transcription takes place. Very much sorry, transcription. After the transcription, the RNA is formed. From the RNA, the sequence is taken, and that is attached to the tracer RNA, and Cas9 comes and takes that as the what we say guided RNA and the tracer RNA both combines into the Cas9 and then fused, and that is what the complex called CRISPR-Cas9. So cutting side, Cas9 makes a blunt-ended double-stranded break exactly three base pairs upstream before the PAM sequence. So coming to Cas9 domains, there are different domains in the Cas9, that is HNH domain, RUVC domain. Repair mechanisms are once the DNA is cut, the cell tries to repair in two ways, that is non-homologous engineering and homology directed repair. It is very much the same that you read in the molecular biology. And Jennifer Doudna and Emilio Carpenter in 2020 have been awarded Nobel Prize for this CRISPR-Cas9 system for developing this method. And when coming to types, there are different types of Cas proteins, that is Cas9, Cas12, and Cas13. You can see in the image. So basically, the Cas9 is used in the gene editing, like nucleic acid detection. Cas12 is used in gene editing nucleic acid detection, but Cas13 is used in RNA knockdown nucleic acid detection. So the target of Cas9 and Cas12 is DNA, but in Cas13 it is RNA, that is single stranded RNA or RNA. Okay, so you have to remember these main points. Okay, so this is the final what we say concept we have read. So I would like to tell you that complete part B rapid revision series has been started and all the videos have been posted. So follow and subscribe to our channel. Join the Telegram group to get instant update. Links are also provided in the description.